Science video. This video is all about mutations. First of all then, errors. A gene is a small section of DNA that is found on a chromosome. Each gene codes for a particular sequence of amino acids, which are then used to form a specific protein. Sometimes these genes may mutate. A mutation then is a random change in an organism's DNA. These can occur when a chromosome doesn't replicate properly and can change the sequence of bases in a gene. There are certain risk factors. Mutations occur all the time, but the chances of them can be increased by a number of factors. Exposing to radiation, exposure to certain chemicals. There are three types of mutations. The first one is an insertion. This is where a new base is inserted into a sequence of bases. As every three bases code for a particular amino acid, this can affect the protein formed. That is not just one triplet code that is affected, but all the following ones too. For example, the triplet code shown on the left is what it should be like. But if this was changed where the G was inserted, this would affect every code that follows. This is because our bodies will read three letters, three bases called a triplet code. So if it read the first case, it would read TAT, CCG, TAT. But now that it's been affected and the G has been inserted, the TAT would remain the same, but instead of reading CCG, it would now read CGC. Instead of reading TAT, it would now read GTA. As you can see, everything after this G has been affected, so this could have larger consequences. The second type of mutation is a deletion. This is where a base is deleted from a sequence of bases. Again, as every three bases code for a particular amino acid, this can affect the protein formed. That is not just the one triplet code that is affected, but all of the following ones too. So for example, we've got the same code. This time, the C has been removed. So again, the TAT is unaffected, but every following other triplet code is affected. Finally, we have substitutions. This is where a base is substituted for a different base. As every three bases code for a particular amino acid, this will only affect one amino acid, so it might have minimal effect. For example, here you can see the C has been substituted for an A, but the TAT code remains the same. The CCG one is the affected one, but the following code, so the TAT remains the same, etc. Now the effects. Most mutations are harmless. Some though will result in an altered protein, which could have significant changes to its shape. For example, if an enzyme shape is changed enough, its active site will no longer match that of the substrate, and therefore it will no longer be able to bind, and therefore become ineffective.